so we're ready for our last presenter and would like to welcome Oscar. We've asked that you introduce the topic and a little bit about briefly about your bio, but I will put up one prompt. The prompt is this, and I think people know this book, right? Codebreaker, and it is a terrific book that talks about what's going on in the world of gene therapy, and it would be terrific to, if you wanted a primer in it or understand what's going on in the world of CRISPR and RNA. You can't hear? We can hear you.
Now I can hear you. Okay.
Okay. Yeah, I had just shown the audience the Codebreaker book, which was the one by Walter Isaacson on gene therapy and CRISPR and so forth. So it's a very timely topic. And we'd like you to share your experiences with the gene and cellular therapy as it relates to clinical research. So take it away, Oscar.
So thank you so much, Len, for the introduction, and also the team for giving me this opportunity to speak here today. I'm really elated about that. This is a great topic. I think, as you just mentioned, we're moving into the gene and cell therapy kind of new world in biopharmaceuticals, and I'm really happy to have been in this space now for over 10 years. After over 25 years in the industry, I started working with Abbott Labs and AbbVie, working with the Humira team, and as you know, that's a blockbuster standard biologic and anti-TNF, and during all these years, and especially in the last year and a half with ASC Therapeutics, I've been able to realize the potential that gene and cell therapy have, especially in relation to personalized medicine. And let me tell you that the first impression that you get when talking about these type of therapies is that they are already targeted on their own, because we have already a kind of targeted therapies that really are addressing a biological conundrum, a human biology question that is already relatively well understood from the pathophysiological perspective. Now, the reality, in my view, is that we need to do more than that. We need to ensure that we have a number of biomarkers, that we have a way to select patients very thoroughly and then monitor these patients. So the fact that the patient has a monogenic disease, and we're going to be talking today about hemophilia, and specifically hemophilia A, that's where we're working, and also in another disease that is related to bone marrow transplantation. So we think that there is much more than can be done there, and that's what I'm going to be speaking about today. So this is an outline of my presentation. I'm going to start talking about, in general, about personalized medicine in gene and cell therapy clinical trials. I'm going to start giving you some examples of the relevance of biomarkers for patient selection and biomarkers for patient monitoring. And then I'm going to use case studies that I know very well, which are on hemophilia A gene therapy. That's the program that we have at ASC Therapeutics, and also on graft versus host disease and the cells, a type of stem cells, stromal cells that are derived from the disease. So we're going to take a look into these specific kind of cases. So let's start with personalized medicine in gene and cell therapy. And of course, you all know that the standard approach that we've been having, especially with small molecules, and even I would say with biologics, is to ensure that we had the one-size-fits-all approach. And this is, although we know already from our clinical trials that there are a number of patients that are not going to respond. But still, we tell the FDA we got already the statistically significant difference. That means that we can put this patient in with this drug when they have certain type of disease condition. That's not everything, because we know again that certain patients are going to respond and others are not. Now, when we now move into this personalized medicine approach, then the treatment strategy is more based in the patient's unique genetic profile. And depending on the genetic profile of the patient, we are going to have either a targeted therapy, which is going to be adequate for these patients, or we're going to have other patients that cannot take this targeted therapy and should get a standard therapy. So that's a very simple way to define those patients that are receiving these targeted therapies. Now, the reality is much more complex than that. The reality is just that not only you and your genes are responsible for the diseases that you have, also your family health history, that which may include many other specific diseases, conditions, many other factors. And we know that although there are about 100,000 genes that have been associated with many common diseases, from diabetes to heart disease, there are only about 5,000 diseases that we consider rare diseases, monogenic diseases, where we really know which gene is really responsible for the disease. But even in those cases, the patients respond and really develop over time with these diseases in a different way. And that depends on many things that are environmental. And among those environmental factors is the diet. Also, your body changes over time with age. Your body mass, your weight, that drives, and we know that very well, especially when you have infusion, and the way we treat patients with gene therapy depends on what we call vector genomes per kilogram. So we adapt the amount of gene therapy that we're giving to patients depending on their weight, and also other health problems. So in general, we see that the complexity is why we need doctors and why we need to treat patients in the right type of environment, taking many things into account. Now, there is also something that is also happening beyond genomics, which is just understanding the genes. These genes are expressed in different ways depending on many aspects, which are called the epigenetics, that's why we also need epigenomics, we need transcriptomics. There are a number of proteins that obviously are different in each individual, and also the way these proteins phosphorylate and the way they are metabolized. So that's why you have here to the left all these omics. And this is what the FDA, and this is a statement from the FDA, is telling us. We need biomarkers that provide information that is essential for the safe and effective use of a therapeutic product. We know that that's the goal. That's what we try to reach. But it still is going to take some time. And when we interact with the agency, they realize that in the majority of the cases, to demonstrate safety and efficacy, we need to demonstrate that with actually clinical outcomes. That's why the majority of our clinical endpoints are indeed this type of clinical-driven endpoints. But the reality is that in many cases, when we are not able to see that, because sometimes it takes years to see a response. I work, for instance, in rheumatoid arthritis, and that's the case for what is called radiographic progression, destruction of the joints that can be radiographically demonstrated. We need something more than that. So that's why at the end of the day, where we're trending towards, or not, we're not there yet, but we're going in this direction, is into creating signatures. And these signatures are the combination of a single or several biomarkers, and then using either artificial intelligence, the so-called also learning systems, and bioengineering. That's when we get into the real application to different type of patients, the way that we subdivide those patients. And we're seeing that happening in diseases that we thought that were relatively homogeneous, like Duchenne muscular dystrophy, Alzheimer. And I'm just telling about a couple of diseases that I read some articles recently where we know that there are different subtypes of diseases, and they depend on all that, everything that we are here. Again, all this work is absolutely necessary to be able to move forward in terms of personalized medicine. Now, if we were to just combine all the different type of biomarkers and see how they are used in clinical trials now, we will see that, first of all, we need to consider those markers that are telling us which patients are going to be talking in just clinical trials, and I'm a clinical development guy, perspective from in terms of inclusion and exclusion criteria, which patients should be the ones receiving certain type of therapies. And we're going to see some examples of how it works, for instance, with hemophilia A, patients that have a defective factor VIII. So we need first to genotype those target genes. And we know that there are a number of patients that indeed have a clear defect on one of those genes. In other cases, we need to phenotype the target cells. So we are not able to identify the actual defect, but we see how these cells are not functioning as expected. Other ways to do that is to take certain cells or target systems or target organs and ensure that we look into functional effects that we are able to identify ex vivo. That's one example on many kind of immune response and ways to understand how the cells respond when they are exposed, for instance, to an antigen. Once our patients have been selected, that's when we move into monitoring the patient response over time. And this is absolutely critical, because in that way, we're going to be able to assure the maximal patient benefit. And we need to have a number of biomarkers that differentiate the two key aspects of any clinical trial, of any work that we're doing with actual patients, ensuring that our products are efficacious and safe. So we need efficacy biomarkers that are related, in this case, to gene and cell therapy, cell product clinical activity, and we're going to see in a moment. We have F8, which is factor VIII gene, which produce the factor VIII protein, which together with a number of other factors are going to be involved in the coagulation cascade and avoid bleeding of these patients. Or safety biomarkers. Is there anything that potentially could lead, in the case, for instance, of gene therapies, that we are providing excessive amount of a certain gene, and it could even lead to unexpected adverse events. Now, let's start diving into examples that obviously I feel very comfortable talking about, because this is what we're working on. And this is, as I mentioned a moment ago, we are now at a completely different process and progress in terms of where we are as the biopharmaceutical industry in relation to where we are years ago, when we started working on it. Small molecules, even large molecules, large biologics. On one of them is Humira, adalimumab, or anti-TNF biologic. We started working on them, pegylating them, but where we are now is in creating an extraordinarily complex type of therapy, which is very large, biological. It includes several components. It is usually a virus that has been manipulated, includes the information that is going to be inserted into the target cells. And as you can see here at the bottom, the complexity is tremendous. And I remember when I was working with the jet fighters, which was Humira, an anti-TNF, a biologic, producing CHO cells, and I was working, I went a couple of times to see our bioreactors in Puerto Rico from Abbott AV, and now it's incredible the amount of complexity that we have when we are able to put together these gene therapy vectors that are, as you can see here, a kind of international space station. There are so many elements that really need to work well to be able to really have the patient's impact. And by the way, is one of the areas that I was not supposed to be involved, and I never even thought I could be involved with, is manufacturing. But now I am. So it's part of my role as Chief Medical Officer of ASC Therapeutics to ensure that I understand how the product is going through the different process of manufacturing, what we call upstream, which is first kind of growing these viruses and then purifying them and then ensuring that they're sent to the clinical side in a way that they can be frozen, thawed. So all these things I am now involved, and I didn't need to 10, 20 years ago. Okay. So let's go into hemophilia A. Is a very well-known disease. Hemophilia is when, again, the blood is not able to clot because of having a factor missing. In the case that we have factor 8, we're talking about hemophilia A. If it's factor 9, we're talking about hemophilia B. This is a monogenic disease affecting exclusively the X chromosome, and therefore it affects only males. And there is also the possibility, so that's why it's very important, that there are a number of antibodies that are inhibitory. So some people with hemophilia, because they have received so many transfusions, or transfusions, or they have been receiving recombinant factor 8, they have developed inhibitory antibodies. These patients right now cannot be served with gene therapy. So, but still, we have a number of patients. That's why the opportunity is very large, and we can still help patients. I just want to give you a sense about the way BioMarin, which is a company also working in this space, recently made some analysis in terms of health economics, and they assessed and they identified a number of an approximate dollar figure of what these patients are costing to the healthcare system through their entire life, and this is about $25 million. So that's why when we talk about these therapies being expensive, and even the production of the drugs can run around $1 million per patient per infusion, and these patients only require infusion. You can imagine that we're talking here about big numbers, extraordinary complexity, but that makes it worthwhile because of what we are saving in terms of helping these patients. So in the case of being able to produce or synthesize factor 8, the most important aspect is to identify what is called a cellular biofactory. And in the case of the type of viral capsid, or the virus that we use as a vector, and the transgene, the gene that we put inside, our target is the liver. So in the liver is where we have our virus getting inside the cell, in the endosome, and then starting to get into the nucleus, but not being inserted into the genome. And we're going to see how that works. So the DNA does the process, and it's very important, therefore, and I always say, now that we're getting ready and preparing our clinical trial, that we need a pristine liver. We need someone that is able to have a liver that is able to produce this protein. And you may think, okay, factor 8, yeah, it's something that is produced by the liver. It is, but it's not produced by the hepatocyte. So the hepatocyte is not used to produce factor 8, and especially the type of factor 8 that we're giving these cells, because we need to manipulate the factor 8 to be able to first be packed into the virus and then being delivered to the hepatocyte. So here is the way I like to use an analogy, which is the historic travel from the Earth to the Moon, where Apollo 11 had a number of components that will help me now to explain to you how we needed a rocket, we needed a promoter, a capsule, we needed a lunar module, and this lunar module either landed in the moon or was orbiting around the moon. And you're going to see what I mean by that. So first of all, we need to make sure that we have the rocket and we have the combination of a promoter and the lunar module. That means that we need a virus, and the virus that we use is adeno-associated virus. It's a harmless virus, and is the one that is going to incorporate the gene that we want to really produce to provide to the liver cell, and the liver cell is going to synthesize and release. The one that we are using in ASC Therapeutics is a second generation transgene, because it includes 91% human, 9% porcine. Why that? Because we licensed this product from the Emory University. They had been working on the phylogenetics of factor VIII for almost 20 years. They developed Ovisur, which is 100% porcine product of factor VIII. And we have seen in our work that this combination of a specific promoter and this transgene, which has also part of the gene deleted in order to make it fit inside the AAV8, that's what we have as a unique product that differentiates us from other companies working also in the same space with a regular fully human 100% product. But everyone does what we are doing here, which is a B domain deleted. You always need to delete a part of the factor VIII. And as you can see to the bottom upper right, we are not incorporating this gene into the chromosome. So it's orbiting around the moon, which would be the kind of the chromosome. And what we were able to see in our preclinical data that is leading us into the phase 1 clinical trials, we were able to see that the factor VIII protein levels were significantly elevated in dose-dependent manner, higher than the regular only human product, that we're able to see some protein inhibitors in some case, indicating in preclinical data, because they were animals that are not used to see this factor VIII, that they were also to produce these protein inhibitors, antibodies, and there were no adverse events. So when we have all this information, it's going to be presented in the next few weeks to the FDA. We hope to get an IND clearance and then start treating patients in the next few months with the product you see here called ASC 618. Now, there's another approach that we can have here, which is using CRISPR. And using CRISPR gene editing, in this case, as you can see in the top right, we are landing in the moon. So we are using the same transgene, which is incorporated into a certain targeted section of close to the albumin locus, and it starts producing factor VIII without using a promoter. So when we do this, we can also treat kids, because with the regular gene therapy, it's not possible. So when we do that, we have the high potency and efficacy and safety. We have a product which is right now in development. It's going to take us some more time to be ready to do this in vivo CRISPR gene editing, which is going to be available for the pediatric population, which is not the case right now for gene therapy. So with all of that, we have. So
Oscar, let me interrupt. We have about 2 minutes left or so.
Okay.
So I'd like to see if you can pull it all together for us, because we have to move on to the next panel.
Okay. Yeah, because we started late. Yeah, I see now. It's about instead of 30 minutes, about 20 minutes. That's fine. So I'm going to skip then the cell therapy section, and I'm just going to end up here with the fact that both in gene therapy and CRISPR gene editing, we will need to first identify the right population, have the selection biomarkers, which are either these antibodies or neutralizing antibodies to the virus, not to the protein. It's very important to make sure that we have liver function tests. We identify factor VIII mutations, because depending on them, the patients are going to respond differently. And then these monitoring biomarkers, either looking into safety or efficacy aspect. And I'm going to skip the cell therapy section, and I'm going to end up just telling you where are we moving towards. The future drivers of personalized medicine in gene and cell therapy clinical trials should improve the understanding of the biological underpinnings of genetic drivers of disease. So it's a chicken and an egg. We are understanding the pathophysiology of certain of these monogenic diseases, and also thanks to gene therapy, we're going to understand also much better how these diseases work. Thanks to next generation sequencing and computational biology, this will allow us to understand better and discover new disease mechanisms. And then, of course, if we really want to have predictive analytics, if we really want to have the real objective of personalized medicine to many individuals, we will definitely need big data and machine learning and a number of computational aspects that are going to help us do that. With that, I thank you, everyone, for your attention. And if there's some time for any questions, happy to take them. Otherwise, thank you anyway.
So, Oscar, again, thank you. I found it fascinating to actually see it in practice. And I don't see any questions in the queue, but, like you said, with AI, with physics, with other things, electron microscopy, all these things giving us more and more insights at the cellular level, the treatments can only get better, as we move ahead. So that's terrific. So Sam, are we going to move the group over to the next panel now?
Yep. Thank you once again, Dr. Oscar. Just in case you guys were not able to catch his talk last time, we do have it recorded on our website and YouTube page, so we can always share that as well. And again, I will be moving everyone to the panel discussion. Thank you.
Thank you.